as it has already been studied for quite a while, and there are well-defined hallmarks of aging by now. So these hallmarks basically describe things that happen during aging, but interestingly, they are also malleable. So we can modify them and change aging. And this is, for example, true for telomere length or epigenetic alterations. Proteostasis is changed with age. Mitochondria, the dysfunction is happening. And it has been shown that all of these things contribute to the aging process and that they can be altered. And my old lab, also Linda's lab, was mainly interested in deregulated nutrient sensing, and that was the main target or the main topic of my PhD work. So the main, or one of the main nutrient sensing pathways is the insulin signaling pathway, and as Carlos already introduced, it's a key regulator of aging. And we know this because we can modify insulin signaling either by diet, by drugs, or by genetic manipulations. And it has been shown by now in several species, as for example in the fruit fly, that if we reduce insulin signaling, this leads to a lifespan extension. And this is an example from our lab where we genetically modified the flies to lose 3 of the insulin-like peptides in the fly. And the survival graph shows basically the survival in black of wild-type flies. They live around 60 days in our lab. But if they lose these insulin-like peptides, they live up to 90 or even 100 days. So we see a quite robust lifespan extension and also health span expansion in these mutant flies. And this has also been shown in other animals. So from yeast, worms, mice, this seems to be a quite conserved finding that if you reduce insulin signaling, this extends health and lifespan. And it has even been shown in primates by dietary interventions. So if you reduce the calorie intake of primates, this also leads to health benefits at old age. So on the left side, you can see the primate that can eat as much as it wants. And the primate on the right-hand side has the same age, but it got 40% less food. And you can already see by the picture, it looks much healthier than its colleague on the left side. So in total, we can say that downregulation of the insulin pathway is really a key player in health and lifespan extension, and it has been studied a lot before, but not so much in the context of circular RNAs. And this is basically where my project started, because it's good now to see also people that publish this work. So the paper that you already mentioned from Mariela in 2018, where she showed that circRNAs increase with age, not only in worms, but this has also been shown in flies and even in mammals. And this increase seems to be, at least in some of these species, happening only in the neuronal tissue. And that was basically when we started my work, and basically to tackle, or to get an idea what could happen with age. Of course, we also kind of need to know what these circuits can do. And for a very long time, as you probably all know, they have been considered to be just splicing byproducts without any function. But by now we know that they can either act as microRNA sponges, and some of them are also translated, and there are also many other functions that have been described. And in total, I would say it's a quite diverse group of RNAs that can have different functions. So coming back to our favorite long-lived insulin mutant fly that I showed you in the beginning, these DIP235 mutants that I showed you, we wanted to use these mutant flies that are long-lived to study circRNAs. And the way we started was with RNA-seq. So we collected tissues of the fly at day 10, 30, and 50, so basically during the aging process. And we dissected the 4 major tissues of the fly, which are the brain, the thorax, which is mainly muscle tissue, the fat body, and the gut as well. And we started, of course, first by verifying published results. So we also first looked at global circRNA accumulation, and we were quite happy to see that also in our data set in the brain, we saw a steady increase of global circRNA levels with age. And also similar or in line with other publications, this did not happen in the other tissues, as for example shown here for the thorax. So now the addition of our work was then, of course, these long-lived insulin mutants, and we were quite excited to see that circular RNAs also accumulate in the long-lived insulin mutants, but to a much lower degree. So this seems to happen slower in our healthier flies, suggesting that the circular RNA accumulation with age is first of all malleable, so you can change it. And second, because these insulin mutant flies are in general healthier, we assume that this accumulation is probably nothing that you would like to have, and it's probably not very beneficial. So after looking at global circRNA levels, we also wanted to look at individual circular RNAs and identify a circular RNA that is differentially regulated in our long-lived insulin mutants. And these volcano plots show a brain tissue, again, the 3 ages, young, middle-aged, and old on the right side. And we found many circular RNAs that are differentially expressed in our insulin mutants. And one that was quite striking or quite, obviously standing out was circSfl, because it was one of the strongest regulated circRNAs, and it was also very consistently upregulated at all ages and also all tissues, which I will also come back to later. So we picked this circular RNA as a candidate to further study its function and hoped we hypothesized that it may itself also influence the aging process. So about the circular RNA at that time point, nothing was known, but of course we know a little bit about the host gene where the circular RNA is made from, and it was called, or it is called sulfateless, and we know that the protein that comes from the linear transcript makes heparan sulfates by sulfation of lignac. There are also some links to misfolded protein response in flies, and we know that it's probably localized to the Golgi. And you can also see the assumed protein structure of this of sulfateless on the right-hand side in this little graph. So basically, there's a cytoplasmic part, so the N-terminus is in the cytoplasm, and there is a transmembrane domain, and the main part of the protein, also enzymatically active part of the protein, is in the lumen of the Golgi. Also, I would like to draw your attention to the locus of sulfateless. So this is a quite simple locus, I would say. Also, even for a fruit fly, it has only 2 linear isoforms, RA and RB. And the only difference between these 2 isoforms is that the second exon, this little 37 base pair exon in the 5' UTR that is the left-hand side here. And so one of these isoforms has that exon, the other one is missing this exon. And also importantly, the circular RNA that we identified in our RNA-seq is actually made by the 3rd exon, which is also circled here. And this exon does not only make a circular RNA, but it's also the first coding exon of the host gene. So there is an ATG start codon in that exon as well. So the first thing we did is we verified our RNA-seq by qPCR. On the top graph, you see circSf-peptide expression in the 4 different tissues that I mentioned in the beginning. In gray is always our white homey wild-type flies, and in red you see our long-lived insulin mutant flies. And we again see very nicely that this circular RNA is really strongly upregulated in all the tissues, up to 7-fold in our insulin mutant flies. And because circular RNA splicing is often linked to canonical splicing, or there's actually some overlap, we also were very interested in the expression of the linear transcripts that come from the same host gene. And on the bottom, on the left-hand side, you can see the expression of sulfateless RA in the same tissues of the same flies. And this isoform does not seem to be changed that much. However, if you look on the right-hand side, you can see that sulfateless RB, which is the isoform without that little exon on the 5' UTR, actually has a quite similar pattern compared to the circRNA. So this also upregulated in all the tissues of the insulin mutant flies. So both of them seem to share some common regulation. So the first thing, of course, we wanted to do is to see what happens if I modify this circle in vivo in the fly. Does this do anything to the fly? Does it make the fly happier? Does it make the fly healthier, or maybe even long-lived? So what we first had to try out is how to overexpress a circular RNA in vivo. And of course, we only wanted to overexpress the circular RNA without the linear host gene. And at that time, this was not done in flies. So that's why we tried different constructs. So you can see the first construct on the left-hand side is basically only overexpressed the circRNA exon and nothing else. And this did not change at all the expression of circSfl. The second construct we tried is the one where we had the circSfl exon plus 1,000 base pairs of the endogenous introns. However, this also did not really increase circSfl expression. And the last construct that we tried, luckily, was one where we engineered the flanking intron. So we basically took the upstream intron, reverse complemented it, and inserted it as a downstream intron. So this basically makes a huge reverse complementary match. And this has already been shown before in cell culture and now also in vivo that this boosts RNA generation in general, and this was also the case now in vivo in the fly in our hands for circSfl. So it was successful, and we got around 100-fold overexpression of this circle in our fly. So then, of course, we were very interested if this does anything to the fly, and I was really super excited when I saw that the flies that have more circSfl that are shown here in the solid line are around 15% longer lived compared to the controls that are in the dashed line here. And this was really quite exciting because this has not been shown for circles before that they can affect the aging of life. And the graph that I'm showing you here is when we overexpress circSfl ubiquitously in the whole fly. However, we also tried this tissue specifically. So we only overexpress circSfl, for example, in the brain or in the neurons in this case, see a lifespan extension in the solid line. However, to a much lesser degree. So the biggest lifespan extension that we got was always with the ubiquitous overexpression of sulfate. So more seems to be more in this case. Exactly. And after that, we then kind of came back to the qPCR of the beginning and wondered what's the link of the circSfl with the splicing of the linear host gene. And I mentioned this little exon that is marked here with the lightnings. That is specific for one of these isoforms, and that was the isoform that correlated also with expression of circSfl. So we decided to use CRISPR and knock this exon out. So basically, we made a mutant fly that only expresses the one isoform of sulfate. And we looked at the flies, 4 different genotypes. Again, white homies are our control flies, the sulfateless exon 2 mutants that I just explained. Again, our favorite long-lived insulin mutant flies in red, and in yellow, purple, the combination of sulfateless exon 2 and the dilp mutants. And we first verified this again on qPCR. So the overall expression of all linear transcripts is not changed in any of these mutants. And in the sulfateless exon 2 mutants, the second and the fourth graph, you can see that sulfateless RA is gone, which is to be expected because we removed that exon. So it was kind of confirming our CRISPR approach. And in the same mutants, sulfateless RV kind of compensated for the expression. So to kind of end up at the same total linear transcript level, RV compensated for RA when it was gone. That was already interesting, but what I liked most was the expression pattern of my circle. So we saw first, if you compare gray to the orange bar on the right-hand side, that the removal of sulfateless exon 2 decreased the expression level of circSfl by around 50%. So it's kind of knocking down the circle as well. And then, as I mentioned, we also looked at our insulin mutants here in red again. And as I showed you before, the circle goes up in these mutants. However, this upregulation is really fully abolished in the double mutants that have a mutation in the insulin peptides, but also in sulfateless exon 2. So that basically tells us that sulfateless exon 2 is not only important for circularization, but also seems to be important or actually necessary for the upregulation of circSfl in our insulin mutant flies. So because of that, we wondered whether the regulation of circSfl and this whole thing done by sulfateless exon 2 is also affecting the phenotypes of our long-lived insulin mutant flies. So this is showing on the next slide. So again, lifespan graph in red are long-lived insulin mutants, in black the controls, and as you can see, the double mutants in purple, they partially abolish the lifespan extension of our insulin mutant flies, suggesting that sulfateless exon 2 and potentially also circSfl are necessary for the lifespan extension of the dilp 235 mutant flies. So it's kind of required. And then, of course, what was still missing, I mean, now we know, okay, the circle is doing something to the lifespan. We can increase the lifespan, we can decrease the lifespan if we modify the circle, but we still didn't really know what it's doing there. And of course, we tested the obvious things. Is it a microRNA sponge? No, we didn't really find any enrichment there. There was nothing very obvious, but then we saw that the exon that makes the circular RNA is the first coding exon of the host gene. And therefore, of course, it also has an ATG start codon, which is highlighted here. And then I got even more excited when I saw that directly after the circle-specific backsplice junction, there's an in-frame subcodon. So this circle basically has everything that you need to be translated. It has a full open reading frame. And this stop codon that I just mentioned is also highly conserved. So also in other Drosophila species, this stop codon is there, suggesting that it could be functional indeed. So we hypothesized that our circle would be translated, and if we compare it now to the protein that is made by the host gene, it actually looks quite similar. So it shares the same cytoplasmic domain. It also has the full transmembrane domain, and directly after the transmembrane domain, when it basically ends, there is this stop codon that is specific for the circle. So if it's translated, it would probably lead to a similar protein, but which is truncated. So then, of course, we wanted to experimentally test if in my circle is really translated. And I again used CRISPR to knock in a flag tag to the endogenous sulfateless locus. And this should, in principle, tag both proteins. First of all, the full-length protein that comes from the linear transcript, but also the smaller protein that comes from potentially from the circular RNA. And this is the plot that I did with these flies. One of my favorite plots I ever did in my PhD. So the wild-type flies are shown on the left-hand side. We don't see any flex protein there, but on the right-hand side, you see flex sulfateless mutant flies. And we see two specific bands, which is exactly what we predicted. So one is at around 100 kilodalton, which corresponds to the size of the full-length sulfateless protein. And we saw a second fainter band at around 25 kilodalton, which is exactly the size of the open reading frame of my circular RNA. So we showed with this that the circle is potentially translated. And then, of course, we wondered whether the circle, if it's the circle, is also regulated in the same way as the circular RNA on RNA level, as I showed you before on qPCR and on the RNA-seq. So I again used Western blot with these flies, and I combined them with my insulin mutant flies. And what we saw here, very similar to our qPCR data, is that also the putative circRNA protein is upregulated in our dilp 25 mutant flies, suggesting that it's not only translated, but it could also be the protein that actually does something to insulin signaling, because it's regulated like it's circRNA on RNA level. And then, of course, the final experiment to prove that really the circle is translated and that the circle protein is the one that is responsible for the lifespan extension that we saw earlier in my circle mutant flies, I made another fly line that overexpresses only the open reading frame of the circRNA. So this is not a circular construct, it's linear, but it's only the circRNA specific open reading frame. And I was very excited to see that also here in the solid green line, I see a lifespan extension compared to the control lines in dashed, suggesting that the circle, if it's really translated, probably mediates the lifespan extension by being a protein. So it's probably not the RNA that does something, it's rather the protein that seems to do something here. And as I mentioned before, I assumed that the protein looks like this with this transmembrane domain. And it seems like it looks, at least from the N-terminals, identical to the host gene. So that's why we wondered, what about the host protein? Does it also do something to the lifespan of our flies? And that's why I did a very similar set of experiments with the linear sulfateless transcript. So I basically overexpress the full-length protein that comes from the linear transcript. And I used the same driver lines as before. I used one on the left-hand side, which expresses my protein ubiquitously in the whole fly. However, in contrast to my circle, this treatment shortened the lifespan. So this was clearly not good for the flies. However, if you look at the brain again, or at the neuron-specific overexpression of sulfateless, I also got a very nice lifespan extension of around 15%, suggesting that not only circSfl-peptide, but also the sulfateless, the classical protein basically, can extend the lifespan of fruit flies. But there seem to be some differences, but also some similarities, because we do see tissue and dose-dependent differences in these flies, but we hypothesize that they may share some, but not all functions, because also only part of the protein is identical. There's also a part that looks different. So the final experiment that we did was to look at downstream signaling of sulfateless. And I'm talking here about the sulfateless host gene, or the sulfateless canonical full-length protein, because about this we know a little bit more. And I mentioned already that it is involved in the synthesis of heparan sulfate. And this is a post-translational modification, and in Drosophila there are only 4 proteins that can, or that basically have heparan sulfates attached to them. So silicon, DALI-like, and perlecan. So now I wanted to test which of these proteins is the downstream mediator of sulfateless that is responsible for the lifespan extension. And that's and this time I not only overexpressed the sulfateless protein in light blue again, but I also overexpressed DALI-like, silicon, and DALI, which is here in red in the first graph, in orange solid line in the second graph, and green solid bar in the last graph. And I saw that if I overexpress DALI-like or silicon, not too much happens to lifespan extension. However, if I overexpress DALI, this leads to a very similar expression of sulfateless, suggesting that potentially DALI is the one mediating the lifespan extension of sulfateless. And then the last thing was the genetic epistasis experiment. So we basically did it the other way around. I overexpressed again sulfateless in light blue. This again extends the lifespan. However, if I combine these splice with a knockdown of DALI by RNA, which is shown in red, this again reduces the lifespan of these splice. So it rescues the phenotype of the sulfateless mutants, really proving that DALI seems to be the downstream target of sulfateless that is responsible for the lifespan extension of sulfateless full-length protein. So as a summary, I give you the graphical abstract of my paper, which basically is that we started with RNA-seq in different tissues of fly during aging. And we showed first that circRNAs increase with age, as shown by others, and that this increase with age is reduced in our long-lived insulin flies. And apart from this global accumulation, we also looked at this one individual circRNA, circSfl, which also goes up in our insulin mutants. And if we only overexpress that circle, we can also increase the lifespan of fruit flies. And the other way around, if we do that trick with sulfateless exon 2 and we reduce the sulfateless levels, we can also decrease the lifespan of insulin mutant flies. And we hypothesized, or actually also showed that this is probably mediated by translation of the circle, because there is a conserved open reading frame encoded by the circular RNA. It probably leads to a protein that is a truncated version of the full-length protein. And we showed that heparan sulfate and DALI signaling are important for the lifespan extension of sulfateless. And with this, I come to my Outlook slide. Of course, there are always open questions and more stuff to do. So we were really interested, what's the shared function between these two proteins? And because they are at least partially identical, we think that could be that they share some interaction partners. So what we want to do is do a co-op here of both protein sulfateless and circSfl, and hopefully find an overlap that could kind of show us what's the important protein that is interacting with both of them. Then this whole story about heparan sulfate is also something that we got quite interested in, because it's not actually linked to longevity so far. So it could also be interesting to measure this in also other longevity mutants. Of course, we also found other circular RNAs in our initial RNA-seq experiments, which we have not too much followed up yet. So there could be also potentially more circRNAs that contribute to aging. And of course, it's always interesting to have a look if these things are conserved. I mean, flies are great, but it will also be good to show this also in other organisms like mammals. And of course, it would be super interesting to see if this function is conserved. So the sulfateless gene itself is conserved. There are actually four proteins that basically are the corresponding genes in mammals, and they all make circular RNAs. They actually make several circular RNAs, so it seems to be a lot more complicated in mammals than in flies. But yeah, maybe someone else follows this up, because as Carlos mentioned, I left the lab after my PhD. So finally, acknowledgements. Of course, a huge acknowledgement to my lab, the Partridge lab. There were several people being involved in this. The Dietrich lab started the whole idea. This is a bioinformaticians lab, and they got interested about circular RNAs, and they kind of initiated the whole project. And I got a lot of support from the core facilities at our institute. And on the right side, you also see a picture of our institute. It's a really fun place to work at. So if you're interested in aging, I can highly recommend that. And yeah, that's all from my side. I'm very happy to take any questions that you might have.
Thank you very much for your talk. And okay, so everyone, if you want to ask a question, you can ask yourself, or otherwise you can write the question in the chat. And if you want to ask, just write a number from 1, 2, 3, 4 in the order you the numbers keep appearing. So yeah, so if you want to ask, we have 10 minutes of questions. So we have one question already from Alexander van der Linden, and the question is: expression of circSfl looks more abundant at 10 days compared to older flies in the volcano plots. At least looking at it quickly, any reason for lower abundance of circSfl at older ages?
Yes, so we actually did that. We verified this by qPCR, and I hope I can show you this. So I mentioned before that most circles accumulate with age. This was actually not really the case for circSfl. So the first graph here is exactly what you just asked, basically what happens with circSfl during aging, so the 10, 30, and 50, and again in our water flies and dilp mutant flies. So we can learn two things from that plot. First of all, circSfl itself seems to, well, if at all, maybe a little bit accumulate, but actually not very much compared to other circles. But this upregulation in the insulin mutant flies actually stays the same at all ages. So there's not really much difference, as you can see in that experiment here. It's always a fold in this experiment, and there doesn't seem to be any change, at least between 10 and 50 days.
Carina, hello.
Hello.
Hello. Great talk and great story.
Thank you.
Yeah, so as it will become clear from my talk, I'm not an expert in circRNA, so I have some general questions. So what is known about degradation of circular RNAs? How their homeostasis is like regulated?
Yeah, I think there are many circRNAs researchers that would like to know that, including me. So there are several reports that report that circRNAs are super stable compared to linear RNAs, obviously, because they cannot be targeted by exonucleases. So if at all, it would be an endonuclease or maybe another a mechanism, but at least in flies, I'm pretty confident that there's nothing known. Yeah, so for me, it's also still a black box. What happens to these circuits? Are they degraded at all, or is this like a specific degradation, or just happening by chance with time? I really cannot answer this question. But I think there is isn't it your talk that talks about degradation of non-coding RNAs?
Right.
So
yeah.
maybe you can tell us a little bit more.
Yeah, well, but it's probably it's going to be some sort of conserved mechanism, but I was wondering if there was something specific about it. And if I can go on, I have a couple of other questions, but still general. But when you mentioned before that you use the sequencing, the flanking sequencing in a palindrome, so what's in the intronic sequence that makes it happen?
So again, this is quite well understood in humans or mammals, but not so much in Drosophila. So I can tell you a little bit about the situation in mammals. So there, it seems like there are ALU repeats in the flanking introns that kind of match. So there's basically matching ALU repeats in the upstream and downstream intron. And they seem to bring together the introns and thereby promoting this whole circularization process, because it brings the ends of the exons in close proximity. Right. And for Drosophila, people have tried to identify motives, but they, at least to my knowledge, there was no clear pattern. So I mean, in my story, I could show that it seems to be some sort of, interaction with the introns, but there's at least no known motive in Drosophila as compared to mammals. Okay.
And finally, and then I'll leave space for other people. Well, do you guys have an idea what could be an interactor of exon 2?
Not really. So there are some proteins that are known that stimulate or reduce circularization. Okay. And many of them actually also interfere with the flanking introns again. But again, not so much known in Drosophila, so it was a bit hard to dig a bit deeper into this. So if I would have had more time, maybe it would also have been cool to pull down these parts of the RNA and see what binds there, because it's really not clear to me what could it be. But it could be indeed something that regulates the splicing of the circle, although this exon is not part of the circle. That was actually so surprising to me that it was so far away from the exon that actually makes the circle, but it seems to do something.
Thank you.
You're welcome.
We have a question in the chat also. What's DALI and is it evolutionary conserved?
DALI is conserved, but I forgot the name. So it is a proteoaminoglycan. So it's one of these proteins that gets a heparan sulfate attached, and its main role seems to be in wingless signaling, or actually many developmental processes as well. So again, this is something that has not been studied so much in the aging context, but we know that a lot of these developmental signaling pathways like wingless or hedgehog, they are all basically involved in this. And that would also be very interesting to figure out in the future, I think.
Thank you. And we have another question. So in here, the question is if you have looked into the integrity of the gut in these age flies and the levels of rot, and if the integrity change in long-lived flies when you overexpress the circRNA.
That's a good question. That was actually one of the reviewer questions as well. So it's a good point, because the gut, the aging gut is a major contributor to the lifespan extension in insulin mutant flies, and especially the gut immunity. So what I looked into in my project was I tried to overexpress circSfl-peptide not only in the whole fly or in the brain, what I showed you today, but I also overexpressed it in the gut only. And there I did not see a lifespan extension. So from that experiment, we concluded that the gut is probably not the major contributor to the effect that we saw here. And that's also why we did not follow up anything in the gut. So there are many people in my old lab that were interested in the gut, but yeah, I never touched on that because we never had any indication that the gut does play a role here. Yeah.
Okay, so thank you very much for your presentation, and now we have to move on to the next talk, and Mariela is going to introduce this talk.
Yeah, I mean, I would like to ask a question, maybe one last question.
Yeah, of course.
I was just wondering, because I find so interesting how the circle just doesn't continue amplifying, like when it produces a protein, like it doesn't do rolling circle amplification like a very large protein. I was wondering if you found first like an iris or something that was the motif that it makes that could make the ribosome to bind and start translating the circle. And second, why you don't see this circle amplification?
Yeah, but the first thing, I have not found an iris. So we also didn't test it experimentally. We rather looked at motifs, and we didn't find anything striking there. The second question was, again, sorry, what? The rolling circle. Yeah. So that's a good question, because I also saw many talks about circular RNAs, and they always reported this rolling circle amplification and these several bands on their blood. But often, and correct me if I'm wrong, but often these constructs were overexpressing constructs. And the Western blot that I showed you was actually a knock-in into the endogenous gene. So this was not an artificial overexpression. So I could imagine that maybe this makes a difference the way, yeah, basically if it's an endogenous or an exogenous like RNA, but just a hypothesis.